Hi, I am Dr. P.R. Padma, Professor, Department of Biochemistry, Biotechnology and Bioinformatics at the Avinashalingam University for Women, Coimbatore, and I shall be your instructor for this module. You have now chosen to learn about the various issues concerning the safety and the ethical aspects that are key to the ongoing debate about whether or not transgenic organisms and their products are safe for consumption. This module is part of the food biotechnology paper under the subject of food science and nutrition. We shall consider the issues that the layman as well as the scientist considers as information that the consumer should be aware of when eating food or a food component that has been genetically modified or has undergone transgenic changes. Such foods are popularly called GM foods or genetically modified foods. At the end of this module, we would have considered the major points that support as well as oppose the use of GMOs or genetically modified organisms, which could be plants or animals, as food or sources of food components. You will be able to weigh the options by yourself and decide on whether or not GMOs deserve to be included in the food that we consume. This module deals with all the major safety and ethical issues which have been part of the global debate between the genetic engineers and the critics who are opposing the use of GMOs have been expressing from time to time. These issues will give you a clear overview of the safety aspects of such transgenic organisms and let you make your own decision whether the advantages of GMOs outweigh their disadvantages or not. Before going into the issues concerned, let us quickly recollect some of the key points of the previous modules, where we have considered the methods of insertion and expression of new genes in the host organisms, be they plants or animals, and their major applications in the field of food technology and nutrition. As we have already seen, the genetic modification or GM involves moving genetic material or DNA from the cells of one organism to those of another, be they related or unrelated species. The resultant organism is referred to as a transgenic organism, or more popularly GMO or genetically modified organism in the common man's lingo. Accordingly, scientists have developed GM plants and GM animals, both of which are poised to play major roles in the areas of nutrition, medicine and health, besides having several other industrial applications and environmental impacts. Our focus will be on the GMOs used as food, providing nutrition and taking care of the health and well-being of the consumer. GM is possible because DNA and the way that genes work are universal in whatever be the type of cell that you are considering. This makes it possible for the inserted gene to express itself and produce the necessary gene product, which is usually the protein coded by the gene. We have already considered the components of a transgene in an earlier module. To recollect this, the newly inserted sequence that leads to a transgenic or GM organism is termed a transgene. For successful integration and expression, a transgene should possess some specific components, namely a promoter sequence for correct expression, necessary sequence modification for better expression or which is the gene of interest, a termination sequence to signal the end of the gene, and a selectable marker gene to identify transformed cells or tissue for the success of integration. The transgene can be transferred into host cells or tissues using several physical, chemical and biological methods, which we have dealt with in detail in module 32 of this paper. You may revise that module for more detailed information. Now, let us consider the safety aspects of genetically modified organisms. Several issues have been raised regarding the production and the release of GMOs into the food chain as well as into the environment. Risks have been envisaged to the human population, to conventional crop plants as well as to the environment. We shall now consider them one after the other, starting with the issues of risk involved to people. These can be considered under the major heads as listed here: horizontal transfer and antibiotic resistance, eating foreign DNA, effect of the cauliflower mosaic virus promoter, and changed nutrient level due to the inserted gene. Let us first consider the issue of horizontal transfer of genes and the development of antibiotic resistance. The issue of horizontal transfer of foreign genes is especially pertaining to the plant kingdom, where the inserted genes may be transferred by cross-pollination. However, such transfer in the human system can also not be ignored because the human beings consume the food products along with the inserted DNA. This has raised safety issues because though the gene has been consciously inserted into the GMO, the horizontal transfer can land it in an organism where it is not supposed to be present. Scientists are ensuring that this does not happen by improving the transgene construct and the methods of their targeted transfer and delivery. However, there is no denying that the transgenes are equipped with the potential to be transferred, and unless governed by strict monitoring and control measures, they may be found in the human systems where they are not supposed to be present. The second issue is the aesthetics associated with eating foreign DNA. Even though we are consuming the native DNA in the food components, eating the inserted DNA is a matter of concern that the foods derived from any kind of transgenic approach should be clearly labeled as GMOs, so that the consumer has the choice of taking them or not taking them. A closely related issue raised in this regard as a safety issue is the use of a promoter sequence in the transgene. As we know, one of the components in the transgene is a promoter sequence to ensure that the transgene is correctly inserted, integrated and expressed in the host organism. The promoter generally used for this purpose is derived from the cauliflower mosaic virus, abbreviated as CAMV. The safety concern in this regard is that the scientists should ensure that the promoter sequence is not dislocated in the host genome by normal processes like transposition, leading to the expression of the wrong genes in the system. This issue is dealt with to a large extent by integrating the promoter and the coding sequence in a manner that they do not separate in the host organism, but skepticism remains in the mind of some. A similar concern also deals with the marker sequence that usually is associated with antibiotic resistance, which is undesired in a host organism. This issue has also been circumvented to a large extent by the use of marker-free transgene constructs combined with methods of targeted delivery into the tissues of the host organisms. The next ethical concern and safety concern that is raised by nutritionists is the changed levels of nutrients in the transgenic organisms. While the GM attempt is aiming at alleviating deficiency conditions, the issue of hyperaccumulation of certain nutrients like the fat soluble vitamins may lead to conditions like hypervitaminosis and needs to be addressed properly when we are trying to enhance the quality of the transgenic produce. These safety aspects are being addressed by the present attempts of plant as well as animal transgenesis, and in the near future a complete solution to these issues are envisaged. Let us now consider the risks that are projected to the conventional crops by the introduction of transgenic crops. The major focus in this area concerns the crop-to-crop gene flow. A major transgenesis approach that has raised this issue is following the insertion of a sequence of DNA called the terminator sequence in the transgenic plants created by companies like Monsanto. The presence of the terminator sequence ensures that the plant cannot be bred through the F2 generation. The company will benefit from this because the presence of the terminator sequence ensures that the farmer is not able to self-breed the GM crop and is dependent on the company for cultivating the genetically superior GM plant. While this is acceptable to the farmers, the issue of horizontal gene transfer has been projected as a cause of worry because the transfer of the terminator sequence to conventionally bred non-transgenic plants may lead to a loss of the line. Several countries, including India, have banned the cultivation of GM plants that contain the terminator sequence to ensure that this does not happen. The use of GMOs has also raised safety concerns related to the environment. The major issue that was raised in the past also involved the crop to plant gene transfer. The concern was that the transfer of the superior qualities of the transgene to a recipient plant which is a weed by horizontal gene transfer may result in the weeds becoming stronger survivors. Especially of concern is the transfer of the herbicide tolerant trait which can result in the formation of a superweed. This issue is by and large addressed successfully by containing the fields cultivating the GMOs with herbicidal resistance. Several such plants are being cultivated over large areas all over the world, and the issue of creating superweeds has not become a major threat. Another issue that has raised concern is the flow of antibiotic resistance gene sequences to the soil microorganisms and the leakage of the GM proteins into the soil through the roots. The flow of antibiotic resistance to soil microorganisms by seepage raises the concerns that the organisms may become hostile and cause problems. However, in all the successes obtained by the cultivation of GM crops, the issue has not reached the dimensions of a threat and is therefore not considered serious. However, the issue cannot be ignored altogether. However, the issue of the leakage of therapeutic GM products like antibiotics and therapeutic proteins into the soil used for cultivating the GM crops is a matter of concern, as these residues are likely to be taken up by the next batch of crop plants that are cultivated in the same soil. This would mean that drugs which are not meant to be taken by individuals can enter the food chain, leading to complications like the development of resistance. True to this concern, residues of the therapeutic compounds from transgenic plants have been detected in the soil samples taken from the fields. Scientists are now working on solving this problem, and a solution is in the pipeline. We have seen in an earlier module that the conference of pest resistance to crop plants has been achieved by inserting the cry gene from Bacillus thuringiensis. While this is considered a major breakthrough in controlling pest and insect attacks, it is also feared that the insects and pests may develop resistance to the cry product. This will necessitate the spraying of high amounts of pesticide chemicals. This anticipated problem is also being worked upon by scientists, and a solution is expected to be evolved very soon. A similar issue is the conferment of herbicide resistance to plants, wherein the farmers may be tempted to use indiscriminate amount of herbicide to get rid of the weeds infesting the field, because the transgenic plants are safe against the herbicide praise. Education of the farmers cultivating the GM crops has resulted in the alleviation of these problems to a large extent. In spite of these issues, GM foods have been in wide use in the United States right from 1996. Several countries subsequently have also cleared the use of GM foods of plant origin, and the land area involved in their cultivation is on a steady rise in many countries, including India. The same kind of success has not been achieved with the GM animals, because they have not crossed the experimental and clearance stages and reached the market. In due course, when they are cleared for use, we may encounter some more safety and ethical issues which may need to be addressed then. Today, hundreds of millions of people and animals worldwide eat food and feed from GM crops, and there have been no ill effects, supporting fully the responsible approach taken by industry and the regulatory bodies. In the future, GM crops providing extra nutrients or medicinal substances will make a positive and direct contribution to human and animal health. As far as eating foreign DNA from GM food is concerned, we eat DNA every day in our food. DNA is and has always been digested in our stomachs, and DNA from GM plants is no different. However, the concern remains that genes can cross from the food that we eat to the bacteria in our stomachs and GI tract, and these might include antibiotic resistant genes that have been routinely used as marker genes in GM technology. This could render antibiotics ineffective against human and animal diseases. The use of marker-free transgene constructs will efficiently address this issue in the near future. GM crops are more thoroughly tested and evaluated than those from traditional plant breeding programs. GM crops have been tested since the early 1980s in many hundreds of trials and are now grown on more than 100 million hectares around the world annually. However, it should be borne in mind that scientists do not know enough about all the kinds of gene interactions to prevent such harmful effects occurring, and laboratory testing may not detect all the changes. The effects of releasing genetically modified organisms into the environment could, in certain cases, be unpredictable. GM crops have only been released into the environment on a large scale in the last several years. No one knows at this stage what will be the long-term effects. We need to keep an open mind and be alert to the changes. The potential benefits may outweigh the threats, and the relentless efforts by scientists may lead to methods of circumventing all the proposed problems. Potential future GM crops is projected to include vitamin and nutrient-enriched foodstuffs and crops with improved oil profiles that will help reduce coronary diseases, as we have seen in detail in the earlier modules. However, the skeptics who oppose genetic manipulations as a violation of nature-made organisms argue that there is nothing that GM foods can offer that a good basic diet does not already offer. Their views are that manipulating the nutrient level of some foods is not a good solution and could be dangerous. Thus, the debate of the pros and cons of transgenesis continues. You are encouraged to weigh all the options available, right from the benefits offered by the transgenic plants and animals, as well as the risks that are projected against them, along with the efforts that scientists are taking in the earnest to get round or thwart such shortcomings. This will help us to boil down the decision of whether or not GM crops and GM foods will be universally accepted. But the fact remains that they have definitely found a way into our food chain and are making very good impact in the nutritional health and well-being of the population. Thank you.